Good afternoon, everyone. So in today's class, we will discuss about homologous recombination. Okay, and this particular chapter, and especially the event of recombination, has logical and logical implications as well as biological implications, and therefore we have to pay attention to the way things happen in the cell or with the genome. It's important for how you also see the nature of change in the genetic information. So let's get to it. This first sentence of this chapter is nice, and I like it a lot because for several reasons, including saying this is all DNA is recombinant DNA. Okay, all DNA, every DNA molecule that you know of is recombinant DNA. You will encounter this phrase several times or in different spheres of your learning, where recombinant DNA technology and so on. There, when we willfully do it, when we willfully recombine DNA, we call it as recombinant DNA technology. But in nature, recombination happens all the time. Every genome is a recombinant, right? And we will try to first define what is meant by recombination, and then we will see what we have to we'll see the intricate details of homologous recombination. First thing, recombination is, as it says, it's combining of two things, two entities. And here, because we are talking in terms of DNA, we will stick to that. So in that sense, I will draw two DNA molecules and see how recombination can happen. Say this is one double-stranded DNA molecule, and we have another double-stranded DNA molecule, the red one and the blue one. Okay. Recombination can happen in two ways. In one of the ways, it's something like this. Say, in one way, it is that one strand has been exchanged between these two DNA molecules. Okay, the strands have been exchanged. That is one type of recombination. There is also another type of way of recombination, that is mixing of pieces. Something like this is formation of two new DNA molecules by this way, like this. This is also another type of recombination. So I hope you notice the difference between the top one, this one, and the bottom one, right? There are no in the top one here. There are no phosphodiester backbone breakage, right? There is no degradation of any phosphate bonds on the backbones. There is breakage of hydrogen bonds and formation of new hydrogen bonds between these two strands, right? And in terms of in the second category, you see that there is a breakage of phosphodiester bond and formation of new ones. Okay, that is one difference you can think of. Second thing is that the this DNA between, if these two strands are able to base pair, then they should have been complementary. What do you mean by complementary? They should have followed the rules, Chargaff's rules, AT, GC, or Watson-Crick base pairing should have been there. That can only happen if the bases that are present on these strands are in complementary nature. So, for example, A, T, G, C, if this was the sequence, then the complementary strand should have had T, A. I mean, I'm reading it from the wrong way. You should have read it from GCAT, complementary. So first thing we will know is recombination is any type of exchange between them. Exchange of what? Is it matter material exchange or informational? Okay, material exchange or informational exchange. We are talking about genetic information, and all the time we have to remember material is not the problem. It's irrelevant. What is important, what matters is the information. Say for example, I will draw a new one, and I hope I'll be able to convince you. Say there is a DNA molecule ATCAG, and the complementary strand would be TACTC, sorry, GC, right? This is a DNA molecule, and I'm writing another DNA molecule, and it is this, something like this. So the complementary strand will be this way, right? Now assume that this is one DNA molecule and there is another DNA molecule. I'll try to use a different color for ease. So I'll just write it as this is another DNA molecule. Now recombination happened between these two. So what would we get? We will have A, T, C, A, G, and that is one strand from here, and the other strand from second molecule. Say we will call this as the first molecule, and the complementary strand of it would be something like this: T, A, G, T, C. Is there material exchange between these two? Certainly there is some material exchange. I will try to also write the other DNA molecule, the new recombinant in a way, and let's see what happens. T, A, G, T, C. Now we had two DNA molecules, molecule 1 and molecule 2. Now we have recombinant DNA molecules 3 and 4. What has exchanged between these two is matter, right? What was one part of one strand has become a part of another DNA molecule. One strand has been exchanged. Has there been any change in information? No change in information. No change in information. Because we are talking about genes and genetic information, anytime we talk about recombination, it should have been a change in information. Even one base should have been different. If you just look at it, this is one molecule of DNA, the same molecule of DNA. 2 is also what has formed 3 and 4. They also are identical to what is 1 and 2. So there is no change uh, in the genetic information. So you don't really call them recombinants. Okay, but let's just take it. So we had these two types, type 1 and type 2 ways of recombination. So when you want to have strand exchange, we need bases to be complementary, or we should say then we need them to be homologous. Okay, the first way of happening is homologous recombination. When you have exchange of DNA strands, and that is because of homology. Homology is similarity in between these two strands. They should be similar but not identical. If it is identical, as I showed you in the previous slide, then there is no difference at all, so it wouldn't matter anything. In these cases, in the second one, you call all of, there are multiple ways, and you call all of them as non-homologous and non-homologous recombination. Okay, So in non-homologous recombination, there are multiple ways again. One of it is, say, site-specific recombination. Some of them are, they randomly break and sometimes are ligated. And so these are all grouped into non-homologous recombination. But when we read about site-specific recombination and transposition, all those fall under this category. So in this particular lesson, we will deal mostly about the homologous recombination and its implication in biology. There will be some complex phenomena that we will study with specific examples. I would want you to read the textbook and gather as much information, because that is when you will understand the logic of how an organism is able to survive, exist, and reproduce. So I have already described what is meant by homologous recombination. The requirements are, the event is strand exchange. Okay, it doesn't have to be complete strand exchange. Some exchange of strand will be there, and because of which is also dependent on homology. In this sense, homology means similarity. So, or I'll put it as sequence similarity. What is similarity? That means it has so many common features, very highly common features, common sequence or features. You can say sequence, I will put, because we are talking about nucleotide sequence, but not identical. That is important. Okay, you have to remember that it should not be identical. If it is identical, there is no exchange of information. All of recombination we talk in this chapter and the next ones is about change in information. So we will go with some of the some ways of understanding how to resolve DNA and how to look at it as a problem and try to solve it. So there is a scientist named Holiday who observed something like these junctions when he was doing some experiments on DNA. He observed something like these, and since then they were referred to as Holiday junctions. And he proposed model on his based on his research, and that is called as Holiday model. And then we will go to double strand model, double strand break model, and so on. The first is, don't worry if you don't know why is it starting on all these things. The goal of this particular slide, on the two more slides to come, is about understanding how DNA strands can be exchanged within the cell. Okay, that's why we say they are models. Okay, so here are two aligned homologous duplexes. When you say duplexes, it means double-stranded DNA. So here is a DNA molecule, and this is another DNA molecule. They have capital A, capital B, capital C. Those are the genes on that, say, DNA molecule. And in the lower DNA molecule, you have small a, small b, and small c. And now we are going to see how strand exchange or bits of strands can be exchanged between these two DNA molecules. And that is where we are trying to look at as the Holiday model. I hope I'll try to zoom it in, and I hope you can see. So the first one is, sorry. Yes. So if a double strand break happens like this, or for some reason it has happened somewhere within, because of, by virtue of, because of some activity of some proteins, then these things will happen. Double strand breaks can happen, and there is first is aligning these two homologous DNA molecules like this. The second step is, and if there is a break or break is induced by some proteins, there will be processing of those DNA molecules. Processing here is exonuclease activity, so some bits of the DNA are removed. And the goal of this DNA molecule, this processing, is to generate these 3' overhangs. These 3' overhangs. What is the use of that? That is for strand invasion. Before going in there, I want you to also remember we put capital A, capital A, capital B, and capital C on the top DNA molecule, and small a, small b, and small c. So as I was telling, there should be a large stretches of sequence similarity, but should not be identical, right? So here is one part of gene B that is being shown. So the sequence says TTA, okay? On the homologous gene, where the small b, everything is same except there is one base difference. So these two B, capital B and small b are absolutely or very similar but not identical, because there is a base difference in at least one base, like here. So instead of TA, it has CG. So that is the difference between those two. Theoretically, just take it that way. And now what will happen now is strand invasion. There is a 3' overhang that has been generated. That 3' overhang will invade. That is the step called as invasion. It will invade into the other the lower DNA molecule like this, okay? And while it is doing so, it is going to displace the strand that was base pairing with the lower in one of the strands to the other side, okay? And the next step is called as branch migration. You see this is a holiday junction, right? I hope you have I encourage you to watch the videos that are there on the on homologous recombination chapter. You can also go through YouTube videos. They are fantastic for understanding, because this will require kind of three-dimensional visualization. Okay, when we show it in 2 dimensions, maybe your understanding might not be as great. That is where animations come very handy. So the strands have been exchanged. Capital B is base paired with small b here, and capital B here is base paired with a small b. Now what is happening in those sequences? You see here, TTA was the sequence for capital B. And what is with the small b? It is AGT. There is a mismatch between these two, right? T and G. So there is informational exchange between these two strands. Because of homologous recombination, there is a exchange of information. And similarly, in the lower strand, you would see that this is the capital B sequence. It should have been AAT, and the small b sequence is TCA. This is the again a mismatch. Okay. So the consequence of what is going to happen is we are not bothered about it at this stage, because we will try to understand more details and then get in there. There are further steps about these are not at this stage. If you see these DNA molecules, they are not two independent DNA molecules. Okay, so recombination is not yet complete. Only when they become independent DNA molecules like this, then you can call them as call or refer it to as recombination is complete. So let's see the steps just so that we will understand it easier. I put it in straight. First one is alignment. These two are brought in close proximity and they are aligned A with small a, B with small b, C with small c, something like that. The second one is single strand breaks or double strand breaks. Usually now what is more accepted is double strand breaks, so we'll just say double strand breaks. And then we have processing of DNA to generate 3' overhangs, and these 3' overhang will invade the lower DNA molecule. So when it does so, that is called as strand invasion, and it will result in the displacement of the strand to the top DNA molecule and will result in formation of holiday junction. And then the branch, so more and more DNA will keep coming down like this, more and more DNA is displaced upwards. So the branch was here, after a while it will keep on moving, and that step is called as branch migration. And because these DNA molecules are, because of this exchange, they will form heteroduplex, like in this case, which would mean that there is some informational difference or a mismatch, as in this case. Now, the important part of resolution. I have seen your assignments, and I'm actually proud that some of you have done it really well, and so many of you have got it right. So it's a nice thing that you already know some things about it. So what you are seeing is something like this in the after branch migration, and then you have to, for our for resolving, that means the, what is meant by resolving, it means you have to make this entangled DNA molecules into two separate DNA molecules. You are resolving the holiday junctions. So if you twist it like this, and you can if you rearrange it, becomes something like this. It's the same thing. Let's verify if it is same or not. For my understanding, all the time I use a way of trying to see which gene is next to which gene is one thing, and the other part I keep in mind is which strand, say capital A is base paired to capital A here, capital B is base paired to small b here. That is one thing I will follow. So sometimes when you are reorganizing this into this structure, we have to keep those two in mind. One is order of genes, whether it is capital A or small a, or capital B or small b. First one is that. Second one is the base pairing. Sorry. If capital A is base paired with small a, or small b is base paired with small b, or those are the things. So here we have two DNA molecules with a holiday junction here. Okay, I will try to verify what is, compare this structure with this reorganized structure, and I am trying to prove that there is no difference between this one and this one. Capital A, I'm looking at this top strand. So capital A, capital B, small capital C. So here, this is the strand I'm looking at. Capital A, B, and capital C. Now I'm looking at base pairing, right? This one. Capital A is base paired to capital A, capital B with small b, capital C with capital C. Capital A with capital A, capital B with small b, and capital C with capital C. Like that we can do for both these DNA molecules, and you will see that these things are satisfied, which means the DNA is the same, just that it is reorganized. In the animation that is provided, if you go through it properly, you will be able to understand or visualize it easily. Why are we doing this reorganization? It is to be able to resolve these structures. Resolving can happen in two ways, and you should always remember that when you resolve, you should get two DNA molecules which will have genes in this order, A, B, C. It doesn't mean that it has to be small a or capital A and so on. What I mean to say is you should get two independent DNA molecules, both of which are having A, B, C. Okay, you should not get something like, I hope I will be able to make you clear by giving this example. You should not get anything like A, B, B. If you got something like this or so, then it is going to be wrong. That's not what you should get. Okay, when you're resolving, you need to take care of some of those things. Okay, resolution can happen in two directions. One is called as site 1 and the other one is called as site 2. For our for our ease, you can also talk of it as, I'll call this as vertical and you can call this as horizontal. Okay, and then, so if DNA cleavage occurred this way, what will happen? Then capital A, B, and it is joined with this, like this, C, small c. So here is, if you look at this, capital A, capital B, and small c. And what are they base pair to? Capital A, small b, small c. Capital A, small b, small c. What happened to the other DNA? You are starting from here, okay? Capital A, sorry, you'll start here. Capital A, sorry, small a, capital B, capital C. That is what you have here. And what are the base pair to? Small a, small b, and capital C. That is what you see here. That is, now you have two independent DNA molecules. So that is homologous recombination. Okay, you have resolved them. You can also resolve it in on horizontal, or you call that as site 2, and that is if you cut these two DNA molecules and join them up. So here the same way, and what I want you to understand here is something. What is the difference between patch and crossover? There are two words that are indicated here: splice or crossover, and the other one is called as patch or non-crossover. If you have a look at this, the DNA molecule here, I hope you will be able to realize that. One second. Yeah. So splicing is something like this. If you are putting a DNA molecule, if you have a DNA molecule like this, that is splicing, right? Splicing is, the definition or the meaning of splice is to join. If you have broken two DNA molecules and joined something like this, okay, there is a overhang, there is a overlap of, this is the heteroduplex region you can think of. And if you made something like this, that is called splicing or crossover. If you find any molecule like this, that is called patch. You have only a small part of the DNA has come from outside and exchanged to only this much. That is called as patch. Say, if we resolve a holiday junction in site 1, we are likely to get splice regions like this. You can see the gray part is from one DNA molecule and the blue in is from another DNA molecule, right? Whereas compare it with the patch or non-crossover products, is you see that the all DNA molecule is with capital alphabets or genes you can think of, except this small patch of strand, and those are called as patch products. So the same thing, the double strand DNA break repair is much more accepted, or it's very much similar to that of what we are seeing, and we will in nature or in within cells, double strand breaks can occur because of radiation or because of antibiotics or chemotherapeutic agents like bleomycin, which can induce double strand breaks. And it can also happen during replication fork. That we will probably see it in the next class, or I would encourage you to watch it from the animation because it's complicated to visualize it. And it is also It may also happen because of several other reasons. The double strand DNA break model is almost same except about how the what happens with the single strand, how the DNA double strand breaks happen, and how strand invasion and DNA polymerization happens. Let's see that quickly here. So in this case, here you see a double strand break, and it is processing is thought to generate 3' single strand tails or overhangs like this. This is 5', 3'. I also want you to notice one thing here. We always say we should put 5' to 3'. The top strand should always be that, typically. When you're doing homologous recombination, you should see that the lower one is put 3' to 5', and 5' to 3', right? And I also, it will be important if you can keep this in mind while you're trying to do or solve homologous recombination problems, is the top strand, I'll call it as strand 1, and strand 2, I'll call these three, this one strand 3, and this one as strand 4, just for explaining concept. Strand 1 is homologous or exact identical to this strand, strand 4. Strand 2 and strand 3 are identical. Okay, or I will try to put it again. Strand 1 is complementary to strand 3, and strand 2 is complementary to strand 4. Just keep that in mind so that you don't forget how these numbers are there. The polarity is indicated. When you're solving, I would always want that you indicate the polarity very clearly. Okay, double strand break and then processing of the gaps to generate 3' overhangs like this, and then strand invasion. The strand invades in the other DNA molecule, displacing the complementary DNA molecule to this way like this here. Okay, so the second strand invasion is something like this. Because they are complementary, the top strand will base pair with this, and DNA synthesis will start from this 3' overhang based on this DNA molecule. Now, which one is acting as the template? The lower strand, the blue strand, is acting as a template here, and it is going to synthesize the DNA like this. What's happening in the down one? There, similarly, this is a 3' OH, and you have template, so DNA polymerases will keep synthesizing the DNA. And then, so eventually what will happen is, as the branches will migrate, right, these will move this way, and the DNA molecule is synthesizing in this direction, because of which more and more DNA is displaced to the top strand. Eventually what would happen is, at one point, this strand here will bind to the bottom one like this, will come down like this, and it is now generating two holiday junctions. This is one holiday junction, and this is another holiday junction. How and how exactly things happen can differ between event to event, and sometimes we don't understand it completely. There could be many proteins. It all depends. But this is a much more accepted model, that holiday junctions are likely, the double strand break model is likely the most reliable one that happens within the cells, and it generates in two holiday junctions like this, and each of them could be resolved in two ways: in vertical or site 1, or in horizontal site 2. So you can have a resolution of 1 or 2 on site 2, or this one in 1 or site 2. There are several possibilities. So if you get into that, it could have been resolved in 1 and 1, it could be 2 and 2, it could be 1 and 2, and 2 and 1. So you have at least 4 different combinations of how these two holiday junctions could be resolved. So I hope you understood the basic concepts of holiday junction, the double strand DNA break model, and what we will see now is some problems are what we should be seeing, right? Yes, this is where the resolution part is important. You have junction, we can call this, for example, just for reference, we call this holiday junction as X and this holiday junction as Y. So each can be degraded either junction in site 1 or site 2. Before going, I will try to explain a few things in that. I'll quickly draw a holiday junction and I hope you will be able to follow along and try to solve it. You already did that, so it should be fine. Okay. Yes, I'm going to write the names. Gene A, this is B, and I'll put a, instead of C, I'll use E, because you can differentiate between capital B, capital E, and small e. So E, this would be A, B, and E. So this is capital A, small b, capital B, and here is capital E, and here is small e. Now I will do the easiest one first. Okay, let me try to do something else before that. One second. Okay, let that be there. And okay, I'm going to solve this one, and I'm going to solve both. This is X, and this is holiday junction Y. I'm going to solve both in 2, side 2, right? Side 2 is horizontal. So I'm going to solve this as side 2 here, and also this one also at side 2. It's easy and simple because it doesn't have anything to do much. You just have to erase these. You're cutting the inner DNA or something like that. And we always have to also describe what kind of products are being formed. This is where you join them like this. Okay, what kind of products did it form? That is, side 2 is easy. You just break those internals and join them up like that. Let's see what kind of products it forms. You see it? This is all capitals, and the other one is all small ones, right? The same. What kind of products would they then be called as? Um, did we go anywhere wrong? Capital A, small a. Oh, I'm sorry. I made a mistake. Yeah, sorry. This is the whole DNA molecule, right? This is right. And here, the same thing you will see, except that one. You have patch products. You have a patch of exchange of a patch region. So that is a patch product. Okay. Now let's see what happens if it is a different one. I will try to use it somewhere else. I'm going to do a similar one, but now I'm going to do both patch site 1. Site 1 is a little bit complicated, but we will get through it. So site 1 is you're breaking these strands, top ones like this, here and here. Okay, and now we have to join them. I'm going to deviate a little bit so that you'll understand it better. This is joined to that DNA molecule like that, and this is joined to this DNA molecule like this. This one will be joined to this, and this will be joined to the other one like this. So it would get a little bit complicated, but as I told you how to resolve these, you have to follow the order of DNA and also what they are base pairing to. So I'll just, because upon resolution, I will get two DNA molecules. Sorry, this is gone. Okay, this is a DNA and two DNA molecules I can draw. I'll just do it this way. It's easier. Okay. Yes, and now I will try to write the genes. Capital A is continuous with, if I keep following this, okay, I will try to use a different. If I keep following this one, now it is capital A is continuous with small b. Small b is continuous with capital E. And now capital A is base paired with capital A. And this capital A is continuous with capital B, and that capital B is continuous with capital E. Look at the lower strand. Capital small a is continuous with small b. Small b is continuous with small e. And capital A here is base paired with small a is base paired with small a. And the small a is continuous with capital B, and that capital B is continuous with small e. What kind of products did we get? If we see this, we will see that, one second, yes. We'll see that this part of DNA is all capitals, right? Capital alphabets. So there is, again, what you're seeing is you're getting patch products. Okay, those are patch products. So because there is an exchange of these capital B here is the new one that has come to this DNA molecule. Okay, so we can do the same things. What you should have noticed is that if we exchange the DNA molecules, one second, let me check if I can remove. Yeah, if we have resolved these DNA molecules based on, here we did site 2 and site 2. In the other one, in the second one, we have done both on site 1. And what kind of products did we get? We got patch products. When you resolve both of them, both the X and Y holiday junctions with the same site, you will end up getting patch products. We'll see if a different one, and I will do one more, and the others there will be one more left, one more variety that you have to do it by yourselves. And I think this is an important question that usually comes up in exams all the time. Okay, I'm going to solve these two. I'll put it here, X and Y. Okay, this is X and this is Y. I'm going to solve this X with site 1 and dolph and then Y, holiday junction Y in site 2. Okay. What is site 2? As I told, it's very easy. What do? We just erase this part like this. The internal DNA molecules are erased and you just join them. Site 2 is done. It's easy. With site 1, we are going to make cuts on the other part. Where is that? We are going to make a cut here and here. The other strands are broken. And now we have to connect them, and that connection would go something like this, and this part will be joined to this one. So always you follow, that is how I remember, is how to resolve these would be easier if I know what gene is following which gene, and also what is base pairing with which strand, capital or small, and so on. So the first one, we will do it here. It's A. A is continuous with small b, right? And let me use this. And small b is continuous with small e. And capital A is base paired with capital A. Capital A is base paired with capital B. And capital B is continuous with small e. Okay? I hope you remember we solved this for site 2. Now look at the lower strand, A, small a. Small a is continuous with small b. Small b is continuous with capital E. Small a, and then small a is continuous with small b. And wait a second, we have mixed up, or I think we have a little bit of a mixture here. This is wrong. So, sorry. Small a is with capital B, and capital B is continuous with capital E. What kind of products are being formed? If you look at it carefully, you will see that this, sorry, yes, this part of DNA is all capital alphabets, and this part of the DNA is from the other DNA molecule. So they are splice products, right? Splice products are, you call them as crossover products. Okay. I hope you understood how you can resolve. You should, for a practice, you draw this diagram and you can solve it by doing, you can solve X by using, this is X, you can solve this at site 2, and Y, holiday junction, at site 1. And then you will be able to solve these problems very well. I hope you understood how to solve holiday junctions and resolve holiday junctions. This is an um, important part of our chapter, and it involves us. It's something you can solve it, right? Being able to visualize it and solve it. So I hope you would be able to practice more. Always remember about how you want to do this stuff. Then, I mean, like drawing your own rules, like which should um, which should allow you to figure out the answer. Like I said, following which gene is continuous with which gene, and which gene is base pairing with which gene here in this case. Okay, but remember that these are, each is a strand of DNA. So in the next class, what we will discuss is with real examples of what happens. Until whatever you learned here in today's class is mostly about the theoretical part of it. In the sense, what you require is theoretical visualization. So please watch animations and come. I'll quickly review what I have discussed today. Homologous recombination. Every DNA is recombinant DNA. That means recombination is always happening all the time. What is recombination? It could be exchange of strands or it could be exchange of fragments. When you say fragments, it's these two DNA molecules cut and been combined like this. In all these cases, what you should keep in mind is it's not about the material that is being exchanged. What is important is the informational exchange. That is what you should look for. And the recombination, if there are strands exchanged, that means it is dependent on homology. Some degree of similarity or complementarity is required between the participating DNA molecules. In that sense, we would that those will be called as homologous recombination. And in this chapter, we are going to learn about homologous recombination, the ways of how strands can be exchanged between two different DNA molecules or even within the same DNA molecules. So I think you're clear with what is material change and what is material exchange and informational exchange. And homologous recombination has three. We'll go to double strand DNA model. It has three important or four important parts. The first one is double strand break, which could happen by accident, can be induced by some proteins or because of damage. And these double strand DNA's break points can be processed to generate 3' overhangs, and those overhangs, there will be strand invasion into the other strand, and there is a displacement of the top strand to the top DNA molecule. And then DNA replication may take over because there is a gap here, informational gap, that should be filled, and that is taken up by these displaced strands acting as templates. New DNA is synthesized to seal up those gaps, and eventually we will end up with a DNA molecule like this with two holiday junctions like this, right? They should be untangled. If it is not untangled, replication is difficult. You can also have transcription. Many things could be affected because these structures, proteins that are moving on top of it, cannot move through these holiday junctions. So they should be resolved. And resolution can happen in two sites. It's site one. When you're doing it vertical, you're breaking this strand here and the lower strand here. Or it can be on site 2 when you are breaking the internal strands, 3' to 5' direction strands, right? It all depends on how you are organizing. For simplicity, it is better to follow the conventions that are given in the textbook so that we don't confuse ourselves. So there are two types of products: crossover products, or they can be called as splice products, where you have, this is blue one is one DNA particle molecule, and the other one is coming from a different DNA molecule. As opposed to non-crossover products or patch products, where only a bit of this strand has been exchanged between these two DNA molecules. So I thought we have done some examples, and in the next class we will learn about REC-BCD pathway, and some very interesting ones are also coming up with regard to mating type switching in yeast and so on. So there will be a lot of details. It will be a good idea if you at least look at the figures that are being displayed in the textbook and read their legends. Okay, we'll discuss in the next class. Take care and have fun.